Hello and welcome to the WeActually Online Academy. My name is Chris and I'll be your tutor for today. In our previous video we looked at question 1. In this video we look at question 2 of the Cambridge AS Biology paper 2-1 winter 2019, course code 9700. Question 2. Figure 2.1 is a transmission electron micrograph showing the bacterial pathogen that causes tuberculosis, TB. Name the pathogen shown in figure 2.1 that causes TB. Okay, so this should be an easy one mark for you if you can remember the diseases. Try your best to write in italics because, as you know, that's the correct way to name individual species from the binomial system. So it's Mycobacterium. Okay, and there's two ones you can write here. You can write tuberculosis, or you can state bovis. Okay, both are valid marks in the mark scheme, but you must say Mycobacterium tuberculosis or Mycobacterium bovis. Okay. Okay, the next part of the question. The World Health Organization, WHO, introduced a strategy in 2015 to end global TB epidemic. An important part of this strategy is to identify people at risk of becoming infected with TB, use methods to prevent transmission of TB. The BCG vaccination is one method of prevention recommended for use in countries where TB is common. The BCG vaccine contains a non-pathogenic living form of the microorganism that causes TB. Complete table 2.1 using a tick to identify the type of immunity that develops in a person who has been given the BCG vaccination. Okay, well, let's just remind what these options here are. So natural immunity, should we say, comes from you being infected naturally by the pathogen. Okay, in this case, it's a vaccination, so it's an artificial type of immunity. Okay, now it's not a passive immunity because you're not getting it from a mother's antibodies or through the fetal placenta or anything that way. Okay, so it's an artificial active immunity as I am delivering a form of the pathogen into the body, which can then stimulate the primary immune response. So it's artificial and active because the body's producing its own antibodies for the pathogen. Okay, so that's an easy one mark. The rest are all incorrect. So we can look at this question like a normal enzyme question. The idea that it binds to a place that's other than the active site. But remember, when things bind to enzymes, they change the active site, okay? So you could say that as rifampicin binds to RNA polymerase, okay, which is your enzyme, polymerase, okay, it changes the active site shape. Yeah, okay, that's an easy one mark. As you've changed the active site shape, it's no longer complementary, so it's no longer complementary to the substrate. In this case would be the nucleotides, to the nucleotides. Okay, and as RNA polymerase is involved in DNA replication, the idea is that it's effectively stopping the process of transcription. Okay, so it stops process of transcription, and this is because if the enzyme can't bind to the correct nucleotide to start the process of transcription, then, well, there's not going to be any mRNA made. And if there's no mRNA made, then there's not going to be any translation taking place. So this also then stops translation from taking place. Okay, and if translation doesn't take place, then there's no polypeptides being synthesized because there's no mRNA going to the ribosomes for translation. Okay. Some bacteria have developed resistance to rifampicin. However, they are still susceptible to other antibiotics that can be used to treat TB. Multi-drug resistant bacteria have developed resistance to at least 2 drugs, including rifampicin. WHO collects data from all countries on the number of cases of TB caused by rifampicin-resistant bacteria, RRTB, and multi-drug resistant bacteria, MDRTB. Figure 2.1 shows the reported number of cases of TB between 2009 to 2013. Okay, so let's have a look at the question. Okay, describe the trend in the data shown in figure 2.2. Okay, so these questions are always really easy. You just have to tell me what you see. You don't have to explain anything, just tell me what you see. Okay, so as you can see here, from 2009 to 2013, the reported number of cases of RRTB and MDRTB have increased. Okay, so that's the first mark, the general trend. So from 2009 to 2013, okay, the number of reported cases of RRTB and MDRTB have increased. Okay, now what you have to do here is that people make the mistake of just saying the number of cases increases. That's not good enough for any graph. Whenever you describe a graph, always use the title of the y variable of what you're talking about. Okay, this is going to guarantee you the mark. If you use an abbreviation or an assumption, the examiner might penalize you and you get 0. Now the other marks come from using data. So if you work out the scale here, okay, so 10 squares equals 40,000. Okay, so 1 square equals 4,000. Okay, so here you can use data. So in 2009, if you look, you have a total of 52,000 cases in 2009. However, in 2013, you have 136,000. Okay, now you can use some analytical different skills here, so you can work out the difference. So you could do 133,000 subtracted by that to work out the net increase in that period of time. You could also use a percentage to say how much it's increased by as well, by working out percentage change. Okay, that's fine, but whenever you describe a graph, always use data to describe your actual conclusion, what it's actually going to do. Okay, other marks here is identifying a few things, so you can use some other descriptive language here. So as you can see here, that there was very little change between 2010 and 2011. Okay, so that's a potential third marking point. You could also identify here that the biggest increase between all of the years was between 2011 and 2012. As you can see here, it had the biggest increase. Okay, so if you stated that as well, so this is marking point 3, using some other descriptive language other than the general trend. Okay, but this is enough to give you your full marks. 2 marks, don't need to spend very long on it. Just describe it, use data, and mention anything of note in descriptive language. No need to explain anything. Part 3i. Explain how resistance to drugs such as rifampicin actually occurs. Okay, well, this is an easy question, okay? So you can talk about the reasons about why. So let's start with, the most common ones we should know about already. Well, what happens is people are overusing antibiotics. Okay, overusing antibiotics. Okay, some people are actually getting treated for viral infections using antibiotics. So you could say antibiotics, okay, used to treat viral infections. Okay. Okay, so that's one of the marks there. Another thing is that human error. Even if we give people medication, they don't necessarily finish the course of antibiotics, okay? So you can also say that people do not finish antibiotic course, okay? And as a result, any bacteria that have survived can effectively have that resistance present as well. Now, generally, antibiotics and bacterial resistance to them is a process of natural selection. Okay, so this is commonly referred to at GCSE level as well. The idea is that for whatever reason, bacteria maybe developed a mutation. So, developed a mutation, okay, for antibiotic resistance. Okay, this would effectively be a selective pressure then. So this is, a selection selection pressure. Okay. And like all of the things that we mentioned in natural selection, is that the idea is that some of the bacteria survive. Okay, so the bacteria survive. Okay, because they have increased fitness to survive against antibiotic treatment. And as a result, they survive, they reproduce. And as a result, they pass allele for antibiotic resistance, okay, to offspring. Okay, and therefore they can now, all the offspring would have the allele for antibiotic resistance to the drug such as rifampicin. Okay, good. Thanks for watching our video. For a free demo, please register on our website, and I look forward to seeing you in our next video.